Good day, everybody. My name is Olga Igoucheva. I am Associate Professor at the Department of Dermatology and Cutaneous Biology at Thomas Jefferson University in Philadelphia, and today it's sunny and very hot in Philadelphia. I have a long-standing interest in developing various therapeutic approaches, including gene therapy, protein therapy, and the stem cell-based therapy with ultimate goal to treat genetic disorders of skin and muscle. I have been a recipient of Cure CMD Foundation for the past 3.5 years, and I would like to share with you the progress towards developing a new therapeutic modality by use of adult stem cells in two most common subtypes of the congenital muscular dystrophies. I will refer to the CMD, such as Collagen 6 and LAMA2 deficient CMDs, which are both share the same underlying disease mechanism, such as deficiency and the dysfunction of the extracellular matrix proteins. So the Collagen 6 and the LAMA2 related myopathies are very unique among the other hereditary myopathies, and they are often considered as a hybrid disorders with clinical features attributed to both muscle and the connective tissue. And that is why they are also called disorders of myomatrix. As some of you may know, the Collagen 6 CMD represents a clinical continuum ranging from the severe Ullrich congenital muscular dystrophy, or UCMD, and to the mild Bethlehem myopathy. The hallmark of the Collagen 6 CMD is a congenital muscle weakness with the connective tissue abnormalities, including joint contractures and the distal hyperlaxity. Both disorders are caused by mutations in the gene called collagen type 6. It is a protein which is in the present in interstitial connective tissue and located very close to the basement membrane of individual myofibers, as shown this on the cross-section of the muscle from the healthy donor. So mutations in the collagen type 6 could lead to the abnormal functioning of the protein. And as shown on this micrograph of the cross-section of the muscle from the UCMD patient, it can result in very granular pattern. The green, that's the stain for the collagen 6, with dramatic reduction of the amount of the protein in the muscle basal lamina. That's the outer layer of the basement membrane of the individual myofiber, or in the severe cases to the complete loss of the protein in the muscle tissue. So the myrazine deficient congenital muscular dystrophy, which accounts for 30% of the CMD cases, is characterized by the absence or the reduced level of the protein called laminin 211, and primarily due to mutations in one of the chains which are making laminin 211, laminin alpha 2, which is known from the old nomenclature as the myrazine. The hallmark of the LAMA2 CMD is the development also weaknesses in the contractures with the respiratory insufficiency, and similar to the collagen 6 CMD, mutations in the laminin alpha 2 gene could lead to the dramatic reduction of the protein production in the of the normal protein in the muscle tissue to the complete loss of the protein as shown here from the muscle tissue. On the molecular level, the collagen 6 is an extracellular protein, and it's an extracellular matrix protein. It is made of 3 separate genes called COL6A1, A2, and N3, and respectively, each of these chains encodes 3 peptide chains, alpha 1, alpha 2, and the alpha 3. Assembly of the collagen 6 happens inside the cell, and it's very complex process whether all 3 chains, alpha 1, alpha 2, and alpha 3, they undergo through the assembly before being excreted, from the cell to the interstitial, the outside, the cell space, whether they are forming complex network, which is shown right here in red, with the complex of microfibrils in very close association with the basement membrane, which is shown right here, of the muscle cells, and whether the Col6 interacts with several other matrix constituents, as shown here. Some of them, these interactions are physical, and some of them in very close proximity. So similarly, laminin 211 protein is also trimeric protein, and it is composed 3 chains, alpha 2 in red, beta 1 in green, and the gamma 1 in blue. Once assembled, the laminin 211 protein gets secreted also in intracellular space, whether it interacts with other extracellular matrix molecules. And by binding or by interaction, for example, with alpha-dystroglycan, which is here, which is the member of the basement membrane of the muscle cells, and the integrin alpha-7 beta-1, laminin 211 protein protects the muscle fiber from the damage under constant stress of contractions. So in the case when the laminin 211 is not functional due to improper formation, of the protein or the complete loss, that makes the membrane of the muscle become very fragile, and it goes, through the damage and many other unwanted events. So currently, there is no specific treatment for these disabling and life-threatening disorders. Cell therapy, cell-based therapy approaches hold great promise for treating genetic diseases, and they have been utilized in animal models and human clinical trials for different types of the muscular dystrophies, and in particular Duchenne muscular dystrophy. So majority of the studies evaluated ability of stem cells derived or isolated from the muscle itself, as well as, the stem cells from the non-muscle origin to replace damaged muscle fibers. However, transplanted cells showed very limited ability to regenerate the muscle fibers, despite the fact of the production of the therapeutic proteins. So what is the muscle connective tissue, or what we call extracellular matrix? I want you to look, at this cartoon, because we're looking for 2 very special compartments of the muscle called endomysium and the perimysium and the epimysium. It's very important because that's what makes the muscle tissue stay together and provide the nourishment, for the muscle, the tissue, and also it has many mechanical properties, to make, the muscle tissue function normally. So in the perimysium, so what is the perimysium? The perimysium is the space which divides skeletal muscle into very specialized compartments, so-called fascicles. And on this the cross-section of the muscle tissue, you can really see, the fascicles, really, really well. Also, it divides bundles of the muscle fibers, like this one. The perimysium contains very important proteins such as collagen and elastin, which creates the fibers. Also, it has the blood vessels. It has nerves that supply the fascicles. Endomysium, or which is shown right there, where it is, right there. So on the close, you can see that this is the special place which surrounds each muscle fiber and ties adjacent muscle fibers together. So it's right there. It goes everywhere, around, individual myofibers. So what are the cells, which are making extracellular matrix? So the majority of the components of extracellular matrix of the muscle, it's a resident cells which are called fibroblasts. And they have one major function to supply extracellular matrix of the muscle tissue with the proteins such as collagens, elastins, and some other to help the tissue to form functional matrix support. So we truly believe that effective cellular therapy for the extracellular matrix-related CMDs such as COL6 and the LAMA2 rests on the ability of the therapeutic stem cells to supply the normal extracellular matrix proteins such as collagen 6 and the laminin alpha 2 that can prevent muscle cell degeneration rather than on the potential of transplanted stem cells to become the muscle cells. Especially this is emphasized by fact that the collagen 6 and the LAMA2 proteins produced by cells of the matrix, are just the fibroblasts. So if we can deliver our therapeutic cells in intracellular, intramuscular, in the spaces, between individual myofibers, so the cells can make, the proteins such as collagen 6 and the LAMA2 and donate it, to the individual basement membranes of the muscle fibers. So then we need to understand what are the stem cells. So by virtue of the function, there are two distinct types of the stem cells, such as embryonic stem cells and adult stem cells. Embryonic stem cell is responsible for the development of embryo from the single cell and through the multiple rounds of the division, giving rise to specialized embryonic tissues and resulting ultimately in the development of the human fetus. Adult stem cells mostly act as the body's repair mechanism by differentiating or by becoming specialized cells, mainly to replace damaged cells with the healthy ones. So unlike embryonic stem cells, the use of adult stem cells in the research and therapy is not considered to be controversial, as it doesn't involve destroying live embryo. Our body has many reservoirs, of the stem cells. It could be bone marrow, adipose tissue, skin, teeth, liver, muscle. So generally speaking, adult stem cells are necessary to replenish dying cells. And this is especially critical in the tissues with the high cell turnover, such as liver, skin, muscle. So this, or, it can help to regenerate the tissue after damage, of course. So these attributes make adult stem cells most promising source for the treatment regenerative medicine, and the tissue engineering. So historically, adult stem cells were isolated from the bone marrow and subsequently from many other adult tissues, including adipose fat, dental pulp, placenta, umbilical cord, and very recently from the fallopian tube. So many characteristics of adult stem cells make them attractive opinion for the therapy. Just few of them. There is a very simple procedures, to isolate adult stem cells. They have great ability to culture in vitro in very minimal conditions. The most prominent characteristics of the adult stem cells is their plasticity, meaning that it's a potential to become other cell types under exogenous stimuli. For example, they can become cartilage, bone, fat, connective tissue, neurons, lung, and the muscle cells. Also, they have ability to engraft after reintroduction, and some other very unique features. Adult stem cells have been proven success in numerous animal models of the disease and in clinical trials. They avoid the problem with the transplant rejection and the ethical quandary as seen with embryonic stem cells. As of today, there are more than 300 clinical trials on adult stem cell therapies for various conditions, which have been entered in the US NIH clinical trial registry. And the phase 1 data collected from thousands of patients indicate that the use of both autologous and allogeneic adult stem cells appears to be safe. Although bone marrow is the main source for the adult stem cell isolation, so the subcutaneous fat represents alternative repository for the adult stem cells. Adipose-derived stem cells can be isolated by less invasive methods, so usually it's the liposuction. They have relatively lower donor site morbidity and are available in the large quantities for the procurement. So here's a few examples. If you do the one bone marrow aspirate, on average you can get 50,000 of adult stem cells per harvest, but majority of the isolated cells will become nothing but the blood cells. If you collect peripheral blood, you can increase percentage of the adult stem cells, which can become the tissue cells, but the number of those cells is very low per single harvest and approaching about 10,000. So in the contrast, the adipose tissue, or simply fat, can give you on average between 10 and 50 million stem cells per harvest, with almost 95% of the cells with potential to commit to some other cell lineage, and with only 5% of the cells which become only the blood cells. I would like to point out that also adipose-derived stem cells have immunoprivileged behavior and suggesting the potential as immunoprivileged universal cells with the capacity to be used in allogeneic setting and to reduce graft versus host disease. So how adult stem cells from the adipose tissue are isolated? As I said, the standard method of obtaining the fat tissue is a liposuction, and usually the lipoaspirate, or simply put, mechanically process the whole fat tissue, is subjected to the simple enzymatic digestion to help to disrupt the fat tissue to the level of the single cell suspension, followed by simple centrifugation. So as you can see on this cartoon, the small stem cell fraction at the bottom of the tube during the centrifugation gets separated from the mature adipocytes, which represent the highest percentage, of the set of. So the smaller stem cells, which are separated from the mature adipocytes, are collected in the pellet at the bottom of the tube, and this is the cell fraction called stromal vascular fraction can be immediately used for the clinical therapies or be expanded in culture, as shown here. So once these cells are plated, the growing cells from this stromal vascular fraction adhere to the plastic of the culture dish, and those cells which survive are the adipose-derived stem cells. I would like to tell you that adipose stem cells can be isolated not only from the tummy fat. And in fact, our group recently developed a completely new procedure for the isolation of the adipose-derived stem cells from the human neonatal foreskin discarded after newborn circumcision. And of course, we wanted to see whether, those cells, they have similar features, as adipose-derived stem cells isolated from liposuction or from the bone marrow or from some other available, the sources. And our analysis clearly showed that the phenotype of these cells showed the typical profile consistent with the adipose-derived the stem cells as measured by the presence of special molecules, on the surface of the cells. Also, we, of course, wanted to see whether isolated cells are suitable for the therapy of the collagen 6 and the LAMA2 CMD. And for example, on these pictures, you can see that, the cells, the growing, the adipose-derived stem cells isolated from the neonatal subjects are making all three chains of the collagen 6, alpha 1, alpha 2, and the alpha 3. And also, since we want to utilize the cells for the therapy of the LAMA2, we looked, whether those cells are making all three chains for the laminin 211 protein. And as you can see, this is green fluorescence shows clearly that our isolated, established, the primary cells have all three chains of the laminin 211. It's alpha 2, beta 1, and the gamma 1. And also, we look at every other possible level, such as RNA for these genes and some other methods. So as I mentioned at the beginning, the stem cells have very unique ability, or what we call plasticity. So they can contribute under exogenous stimuli, or if you introduce the cells in the tissue, so the microenvironment of the tissue can influence on the cells, forcing them to become the cells with the phenotype of that tissue. And in fact, we wanted to see whether neonatal cells also, have this ability. And using variety of the techniques, we did show that this is the starter cultures, before we start putting any exogenous pressure on them. And you can see that, if you continue to grow the cells in the presence of different combinations of the exogenous stimuli, they can become osteoblasts or adipocytes, or even, the chondrocytes. So the overall This analysis really provided strong evidence that neonatal adipose-derived stem cells represent a cell population with the phenotypic and functional features of adult stem cells, and they produce significant level of the collagen 6 and to a lesser extent of the laminin 2, and are readily available for the therapeutic replacement of the collagen 6 and the laminin 2 proteins in the CMD mouse models. So what are the mouse models for the COL6 and the LAMA2 available to test the therapeutic interventions? The mouse mutant, the lacking alpha-1 chain, or we call it COL6A1- mouse, was generated several years ago by Dr. Paolo Bonalda in Italy, and this mouse served as a model for Ullrich congenital muscular dystrophy. So the phenotype of alpha-1/6 mice is much milder than of recessive UCMD patients with the total absence of the collagen 6 protein. The collagen 6A1- mice have normal lifespan, but are somewhat smaller in size than the wild-type animals, and usually these changes happen later I think usually after 6 months of life. Their skeletal muscle display the myopathic histology, including variations in the muscle fiber diameter and the presence of the muscle fibers with centrally localized nuclei. Physiological assessment of the muscles has demonstrated loss of contractile strength, in that the titanic and the twitch tensions were significantly reduced. Also, myoblasts isolated from the COL6 CMD mice displayed latent mitochondrial dysfunction, or it's a suggestion that that the tissue predisposed to the apoptosis, and the apoptosis is the process when the cell is going through the sudden death. There are 6 different mouse models for the LAMA2 CMD, in which laminin alpha-2 chain expression ranges either from the total loss to the single point mutation. So these mouse mutants mostly recapitulate human disease, and they demonstrate relationship between expression level of the laminin alpha-2 and severity of the disease manifestation. So the gene-targeted DYW mouse strain with markedly reduced expression of laminin alpha-2 chain has been widely utilized by many investigators, including our group. As you can see, affected animals are small, they're passive and emaciated, so they usually have greatly reduced body weight than non-affected litter mates. Muscle of DYW mice contain necrotic fibers, and you can see that the size of the muscle in DYW mice is much smaller. The volume of the muscle is much smaller than in the normal mice, and also analysis of the muscle tissue showed profound, the fibrosis in the muscle tissue. DYW mice are lethal at 10-15 weeks of age, and they display severe muscular dystrophy and the peripheral neuropathy. So altogether, developed mouse models represent ideal genetic targets for the evaluation of various gene therapy approaches. And now it brings me to the data which we obtained during the first 2 years of funding through the QRCMD. And you can, so this work actually is a proof of concept data showing that the human neonatal adipose-derived stem cells delivered intramuscularly can participate in the restoration of the collagen 6 deficiency in the collagen 6A1- mice, and the results of the studies could be downloaded from for free from the Stem Cell Research and the Therapy journal. So in order to, in our transplantation studies, to trace transplanted cells in the animal limbs, the first we labeled our cells with fluorescent chemical compound, which allowed us to visualize transplanted cells in the treated area. And you can see that, that the direct shining of the UV light and on the injected limb helps you to identify with very high precision of the area of the transplantation. And on this zoom micrograph, you can see very nice distribution of the cells, along, the individual, the muscle fibers, and also on the cross sections, you can easily identify cross sections which are containing transplant. After labeling, we transplanted cells intramuscularly in the gastrocnemius muscle, which is right there, of one of the limb of 3-5-old collagen 6 deficient mice. This is the group of our patients. The second limb was injected only with the physiological solution, and basically it served as an internal control for the treatment. So the live imaging of the both the transplanted and the non-transplanted limbs was performed every week for a total of the 6 weeks using special instrument called IVIS, live imaging system. And as shown on this picture, you can see that during the procedure, our patients are comfortably sleeping, they are alive, but it really helps us, to keep them steady, so we can take pictures of the limbs after transplantation. So this is the transplanted limb, and this is the limb which was just injected with the physiological solution. And on the bottom panel, you can see that the red fluorescent signals, these ones, which are common from the labeled transplanted stem cells, were detectable in all transplanted limbs throughout the study, but not in the control limbs. To measure myofibers associated with immune-reactive alpha-1 collagen, meaning that we want to see whether the transplanted muscle tissue has any signal coming after labeling of these muscle tissues, with the antibodies specific for the alpha-1, the collagen, and also to estimate engraftment level, meaning that, whether we want to see how many donor cells survived upon transplantation and what is the distribution of the donor cells inside the tissue. The muscles collected from the transplanted mice every week was cryo-sectioned and stained with the special molecules, as I said, called antibodies, which are specifically recognize alpha-1 in red, and which are representing secreted the collagen 6 by transplanted stem cells, which is completely missing in the recipient mice, and the laminin AC, laminin AC, which is the marker, for the donor cells. As we can see from this consecutive the pictures representing different stages of the study, that analysis revealed the robust, the time-dependent engraftment, we can see the green cells, and the distribution, the red one, of the collagen 6 positive myofibers within endomysial connective tissue, right? The endomysium, that's the space which surrounds muscle fascicles, and also, the perimysial space, and endomysial space, which is it's a space between individual myofibers, and the collagen 6 positive, collagen 6 staining was pretty substantial, and we found many individual myofibers surrounded by the collagen staining. So using additional double staining with antibodies also for the alpha-1 collagen, so this time we used the green color, and another protein called the collagen type 4. This is very important, the protein, because this the collagen 4, it's integral partner, of the BMZ, and it is expressed exclusively at the basement membrane, of the myofibers. So we showed that this double staining resulted in the overlapping fluorescence signals for the both proteins alpha-1 and the collagen 4, as shown here in the yellow, and that clearly suggests that the protein which was derived from the transplanted cells is the functional. So the overall, really, this data indicated that adipose-derived stem cells, they engrafted. They didn't stay in the one place. They travel along interstitial space, between myofibers, and they continued, to produce therapeutic protein into surrounding myofibers. Since muscle phenotype, as I mentioned, and the mouse model description is much milder than the human patients, we took advantage of cardiotoxin injury model to create temporary injury in gastrocnemius muscle of the neonatal mice, thereby exacerbate the muscle abnormality. So the CTX it's a snake venom. It's isolated from Naya, Mozambique, and it's widely used in the research settings to induce the muscle injury. Again, in order to enhance a regenerative response from the transplanted donor cells, at first we transplanted animals with the stem cells, and 2 days later the limbs with injected cells been treated with the cardiotoxin to the induced injury. So as expected, direct injection of a venom into muscle resulted in the severe tissue injury, as you can see here. The histological analysis demonstrated global myofiber fragmentation. 2 weeks the post-injury CTX-treated muscle had numerous clusters of regenerating myofibers, as judged by smaller diameter of the myofibers, basophilic cytoplasm, and centrally located nuclei. But importantly, in the CTX-treated muscle receiving stem cell transplant, the number of the collagen-positive myofibers significantly increased. And you can see that already, at 2 weeks, we could detect really large area of the muscles which stained the positive with the antibodies, the specific on chain. Muscle morphology at 3-6 weeks, the post-CTX injury was not much different, as you can see on the HE stainings, and except that we could detect, the presence of the numerous myofibers with centrally positioned nuclei. It's a known hallmark of the recent muscle regeneration. So functionality again, transplanted cells was confirmed with the collagen alpha-1 antibodies and the collagen 4 showing colocalization, again appearance of the yellow signals. It's a green and red comes together and they give the yellow color. So you can see clearly the colocalization of both COL6 and the COL4 proteins at the basement membrane of individual myofibers. So the overall, this data strongly suggested that the collagen-6 donating adipose-derived stem cells were able to migrate intramuscularly, and we believe it happened in the response to the factors which been released in the course of CTX-induced injury and regeneration, and the cells, migrating cells, continued to produce collagen-6 protein in the muscle of COL6 deficient mice. Now I want to switch the gears and talk about some of the progress which we're making now toward developing stem cell approach, which will allow to address prevention, of the muscle pathologies from the systemical standpoint. Unfortunately, a significant obstacle in the designing stem cell-based therapy for the congenital muscular dystrophies, and I would say other muscular dystrophies and many other systemic disorders, it's the necessity to reach entire body musculature. And that's the problem which cannot be easily overcome unless systemic transplantation protocols are proven to be effective. However, natural homing of the systemically infused adult stem cells to the skeletal muscle is a very inefficient process, as the majority of the transplanted cells get trapped in draining organs such as lung and liver. So basically, we think that the limited homing adult stem cells to the diseased tissue may actually result from an inefficiency of appropriate signals facilitating migration of systemically transplanted cells to that tissue. So what is the cell recruitment? The cell recruitment from blood circulation to the distinct anatomical sites, it's a very complex and regulated by multiple processes, such as recruitment itself, extravasation of circulating cells into damaged tissue, migration of those cells inside the tissue, adhesion of those cells in the tissue, and of course normal functioning of engrafted cells in the new niche. The measure process, which is responsible for cell recruitment in the body, is called chemotaxis. It plays a crucial role in the directing cells, and I would like to stress out especially inflammatory cells during acute damage or chronic inflammation or disease. So it plays a crucial role in directing the cells to distal anatomical sites, and it's highly dependent signaling molecules called chemokines. The chemokines get secreted from the diseased or the damaged tissue. So what happened next? It creates the crosstalk with the cells in the circulation, or we call them rolling cells, and not just with the cells itself, but with the small fragments on the membrane of the cells called receptors. So engagement between chemokines, which are secreted from the tissue, and the receptors on the cells, on the rolling cells, leads to the multiple extracellular events, allowing extravasation of the cells from the circulation and directional migration towards area with the highest concentration of the chemokines, or what we call chemotactic gradient. So what we really need to understand that when we have a disease, or we cut our finger, or we have something, there is any hint of the inflammation, the microenvironment in that tissue changes dramatically, and the tissue starts to secrete these chemoattractant molecules, chemokines, which are engaging with the cells in the blood circulation. And because there is a very strong relationship between chemokines and the receptors, not every chemokine will attract the cell with any receptor. They usually work as a pair. So based on this idea, we are actively searching for the ways to enhance the homing of adult stem cells to the skeletal muscle and possibly to the diaphragm. So here's our current working hypothesis, which is based on the idea that systemically transplanted adult stem cells can be recruited from the circulation to the skeletal muscle and possibly diaphragm via rolling on the muscle vasculature mediated by activation of the specific receptors on the surface of the rolling cells. And we are hoping that this is our systemically transduced the stem cells. So followed by intramuscular migration under influence of the CMV muscle-derived chemokines. So up to date, no sufficient data is available regarding the chemokine expression in the CMV-affected muscle. So to broaden this knowledge, we already conducted comprehensive analysis of chemokines in skeletal muscle of the LAMA2 CMV affected muscle and the diaphragm. So the biopsies of gastrocnemius muscle, diaphragm, and also serum were collected at different stages of the disease. Usually, the initial stage, which is the first couple of weeks during the acute phase, which usually observed starting at week 3, and the terminal, the stage after week 5. This picture shows you representative of this analysis, and each black spot represents individual chemokine. And just by looking the tissue, and this is the time when the tissue was collected from the diseased mice, and at the bottom this is age-matched normal litter mates, meaning that this is the tissue, the same, the gastrocnemius muscle tissue from the mice not affected with disease. And just by comparing, the pairwise, those, the micrographs, you can clearly see that in the DYW tissue, there are numerous of chemokines, or we can call them chemotoxic molecules, which are present at elevated level than in the control mice. So, so the more detailed analysis, or I would say comparative analysis, revealed induction or presence of several chemokine molecules in the CMD-affected muscle, which is right there, and also in the diaphragm of the DYW mice. Functional analysis of these molecules, because each of these chemokine actually, it can tell, it gives you lots of information and the idea about what type of cell can be attracted, to the diseased tissue, and make the level of this chemokine the high or low. So the functional analysis of this identified, the chemotoxic molecules suggested that sustained expression of identified molecules may provide a selective mechanism for attraction of the several inflammatory cells to the CMD-affected muscles, including, but of course not limited to, macrophages, which are right there, the neutrophils, and the T-cell recruitment. So, where are we going from now? Usually, the freshly, and again, if we're going back, to the working hypothesis, so now we know what are the signals coming from the CMD affected the tissue. So now we need to choose the right population of the stem cells which can actually respond to these molecules which are secreted from the CMD-affected tissue. And I want to point out that freshly isolated the adipose-derived stem cells, or analysis of the cells, they showed very limited repertoire of the receptors which are available on the surface of the cells. And for example, on these cartoons you can see that it's very heterogeneous population, meaning that some of the cells, will have this type of the receptors on the surface. Some subset of the cells will have completely different, or something like that. And I think that's what really drives an efficient, not the drives actually, that hampers, the efficient attraction of the cells to the muscle tissue, because we simply don't have enough right cells, to respond or to engage with the molecules which are providing signals, from the diseased tissue. So what we've been doing currently, and actually, we are getting great success, doing it, we are overcoming this limitation that, it's very heterogeneous. So how we can increase or enrich, the population of the stem cells expressing right receptors. So we developed very generalized the selection strategy of adipose-derived stem cells, which can be then, used in future transplantation studies. And of course, we are focusing primarily on the stem cells expressing receptors for the muscle-specific hormone. So that to enrich identified populations, we're using special method which utilizes magnetic separation. And as I said, we have very good results at the moment, which allowing us to really, to get very high percentage of the cells expressing proper receptors for the muscle hormone. So, and at the end, I would like to show you some of the data, whether we've been trying to apply chemotoxic mechanisms for the stem cell attraction or the hormone to the muscle tissue. And in this particular experiment, we've been trying to see whether selected cells with specific receptors have the better ability for the recruitment, to the muscle tissue. In this, the pilot study, so what we actually did, we systemically transplanted adult stem cells through the tail vein of the normal mice. And right after systemic transplantation into mice, what we did, we injected, the chemokine, which is supposed to recruit those cells, to the muscle tissue. On the top panel, this is the mice which been transplanted with cells non-selected, meaning that, they have all variety, of the receptors, and that's how, they are right after isolation. And you can clearly see that, the mice that transplanted with non-selected stem cells showed significant entrapment, which I told you about at the beginning of my talk, in the lungs. And it happens pretty fast, the first 24 hours, majority of the cells stopped in the lungs, and it doesn't go anywhere. And of course, we couldn't detect any engraftment or migration of the cells in the limb, which was treated, with the chemokine, which is supposed to attract those cells. And as you can see in the next, the 2 days, the control mice didn't show any signals even in the lung, indicating the migration of initially entrapped cells in the lungs along unspecific directions or unspecific axes within the body. So let's see, what happened, with the mice which have been transplanted, with the selected cells, the cells which have, the receptor on the surface, which could be very attractive, to the chemokine which is injected, in the muscle. So you can see that in the sharp contrast, the first 24 hours after transplantation, so the migration of the selected cells can be detected in the limb, although, there are plenty of cells, still sitting, entrapped, in lungs. So additional 48 hours led to the more robust migration of the receptor expressing the stem cells from the bloodstream to the chemokine, the treated muscle. And these results are really great, and they greatly support, our original hypothesis that we need to work with the right cell population, and we need to know what are the signals, our tissue sends, so these cells, can be attracted to that tissue. Our future studies, since, the LAMA2 CMD exhibits very severe the muscle phenotype with extensive the processes such as inflammation, fibrosis and the apoptosis compared, to the collagen-6, the CMD mice. We just want to see the effect of the cell transplantation on these pathologies and in more details, and we want to put the special emphasis on the trophic effects of the stem cells in the disease environment. So the trophic effects is that, the stem cells, besides the therapeutic, the proteins which are missing in this, the animal models, they have all other, the good molecules, which they make, which can really, positively contribute to the fight, the inflammation, in the fibrosis. So how, we're going to do it? Mostly, now we focused on intravenous injections of the stem cells, and again, we are not working anymore with the heterogeneous populations since, we know what type of the stem cells we need in respect to the expression of the receptors on the surface. So we are going to transplant only those cells in the lambda2-W mice or collagen-6 alpha-1 mice. And again, because the col6 mice itself don't allow us to make any physiological assessments, those experiments will be done in the conditions of the CTX injury model. And we will be looking at several important criteria during these studies, such as percentage of the transplant, what is the percentage of the transplanted cells become engrafted. This is very crucial because we need to know for how long the cells, can continue to provide, the therapeutic proteins, of how long they can survive in the tissue. Also, we're going to look at the migration of the engrafted cells over the time and their location. Again, are they going to be in the muscle extracellular matrix of the perimysium and the mesonym, or they can become, the cells of that tissue? That's the plasticity, right? Whether, they can become either fibroblasts, which reside in the matrix of the muscles, or they can even become myogenic lineage, such as satellite cells. And the satellite cells, its own muscle, the stem cells. Also, of course, we're going to be looking for the levels of the therapeutic protein production, alteration in extracellular matrix composition, and possibly, we will be looking at some signaling pathways. And the last but not least, of course, the morphological and the functional improvement of the treated muscle. So at the end, I would like to give my special thank you to all patients and the donors. Also, my special thank you to the Cure CMD Foundation and all the staff, at the foundation for your tremendous support. I would like to acknowledge Professor Moon-Lee Chu, who has been instrumental in this project, and she's actually one of the pioneers who cloned the collagen-6 gene. My colleague from the department, Vitaly Alexeyev, and the members of my lab. And I hope, we can talk in the future, and I can show you or present you with more the data, and hopefully, we can fight, this very unpleasant, the disease, and let's all, the bully muscular dystrophy. Thank you all, and have a good day.
Thank you so much, Dr. Igoucheva. We're very encouraged by your progress, and we're very excited to share it with the CMD community. And we appreciate you explaining your work.
Thank you.